Imam, you have the floor if you can offer some prayers, please. Thank you.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الحمد لله رب العالمين الرحمن الرحيم مالك يوم الدين إياك نعبد وإياك نستعين اهدنا الصراط المستقيم صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين الحمد لله هادنا لهذا وما كنا لنهتدي لولا أن هادينا الله الأول والآخر والزاخر والباطن يا ربنا لك الحمد كما ينبغي وجلال وجهك وعظيم سلطانك وصلى الله على سيدنا محمد خاتم النبيين وإمام المرسلين سبحان ربك رب العزة عما يصفون وسلام على المرسلين الحمد لله رب العالمين آمين.
Amen. Shukran, Imam Jallo. Bishop, you have the floor, please.
Thank you, Chairman. Lord God Almighty, God of grace, God of creation, and God who redeems all humankind throughout the whole world. We come before you this morning hour as we continue to do our TRRC sitting. We ask that you will be with us, be with the whole nation, the Gambia, and be with the witnesses that will come forth. And we ask that you will grant that all of them will testify the truth, nothing but the truth. And that, Lord, that you will continue to reconcile this nation, and that the peace will continue to gain its momentum on a daily basis. This we ask through Christ our Lord. Amen.
Amen. Thank you very much, Bishop Wadeko. Council, are we ready with this morning's witness? And I believe we're continuing with the same theme, but from a different perspective, if you would want to just enlighten us on that one. The floor is yours, please, Council.
Good morning, Mr. Chairman, commissioners, and members of the audience. We have a different witness today. We're calling Dr. Assan Jaye, who is an expert in HIV treatment, specializing mainly on immunology. And we're calling Dr. Jaye to help us understand what HIV is, how it was dealt with in Gambia until the time that President Jammeh, former President Jammeh, made his quote-unquote breakthrough declaration, and the impact that had on HIV treatment in the Gambia, and why we find ourselves where we are today. So Dr. Jaye would be one of several other doctors who we expect will testify before the Commission in the next few days. If that is satisfactory, I would ask that Dr. Jaye be asked to come in.
Yes, indeed, that is satisfactory. So he is appearing as an expert witness.
Yes, Mr.
Splendid.
Chairman. Thank you.
Thank you very much. Please proceed.
I, Dr. Assan Jaye.
I, Dr. Assan Jaye,
Do swear that.
do swear that
I'll speak the truth. I'll speak the truth.
I'll speak the truth,
The whole truth.
the whole truth,
And nothing but the truth.
and nothing but the truth.
So help me God.
So help me God.
Thank you.
Dr. Jaye, welcome to the TRRC. And
To be here.
thank you very much for your willingness to appear and testify before this commission. Dr. Jaye, I just want to give you a brief resume of what I expect our conversation today to entail. First of all, we would like to briefly talk about your biography, your work experience, places you've worked, your academic qualifications, your work on the area of HIV/AIDS and your publications. And then we will talk about your work on HIV and AIDS in the Gambia, how HIV/AIDS treatment was approached and dealt with by the MRC in collaboration with the Gambia government. We would also want you to talk about briefly the history of and the discovery of HIV/AIDS, how it was treated in Gambia in 1983, the history of drug discovery of HIV/AIDS, how it was hitherto a death sentence, the discovery of medicines such as lamudivine or zidovudine, excuse me, and lamivudine and all these Dean family, pretty much, the monotherapy drugs, and the move to other more effective drugs such as combuvir or the cocktails as some would call it, the result of those types of therapy, and how ultimately HIV/AIDS moved from a death sentence to a manageable disease like diabetes and others, especially with the new interventions such as the heart treatment, the expenses involved in accessing treatment and how that was solved in Gambia, the initiation of heart treatment in Gambia and some of the ethical issues or some of the protocols that you had to observe, such as counseling, adherence, social and nutritional support, and all that, issues of opportunistic interventions or infections, baseline surveys you would have had to do, and all these things, and how that has led to the results that were registered on HIV treatment in Gambia, especially looking at the cohort of 1,600 HIV-infected persons you worked with in 2005. And what happened when President Jammeh suddenly appeared with a bag of herbs and made his breakthrough pronouncement, how that has affected HIV treatment in the Gambia. But centrally, the criminality in all that conduct, if you would call it as such. You are the expert. We would want to hear from you on that. The impact of Jammeh's claim on the Global Fund and its use in Gambia. We would also want to understand a few things about HIV treatment. The differences between HIV-1 and HIV-2, and how, which one was prevalent in the Gambia and how these things were being dealt with. Basically, these are the things we would want to discuss. How Jammeh's claim has affected the cohort you had at the MRC, the HIV clinic that you had, and how your activities had to move to Senegal, and your own assessment of the PATP. These are the issues I'd like to discuss with you. Quite a lot, huh?
Yes, that's fine. I'm happy to go through all these aspects.
Thank you very much, Dr. Jaye. So, what are your full names?
My name is Assan, A-S-S-A-N. Jay is the surname. Most people confuse it to Gay, but Jay is nicer than Gay. So it's J-A-Y-E, Jay.
I would endorse that, but Dr. Jay, sit comfortably.
Okay.
This may be a marathon session.
All right, that's
so
fine.
Relax and.
I think I have the endurance.
Thank you very much.
Yes.
Thank you very much. Good. So, Dr. Assan Jaye, can you briefly tell us about your educational background?
Okay. Initially, I studied as a veterinarian. I did my veterinary education in Nigeria, where I got my Doctor of Veterinary Medicine degree in 1988. And then at that time, I wondered whether I should actually practice, which was my major ambition, to practice veterinary medicine and become a major farmer, or to be a researcher. But I was convinced by my external examiner to be a researcher. So I luckily got a scholarship immediately to go and do a PhD to specialize in immunology and molecular biology. At that time, it was genetics. And I did my PhD, registered at Brunel University, but I did much of my research work in Kenya. So commuting between Kenya and Kenya and UK. There was an international research center there. So I worked on immunology of trypanosomes, which is a major infection in cattle, and molecular biology, genetics, and made some discovery there for the PhD. Anyway, but after the PhD, I then got recruited, got a fellowship, postdoctoral fellowship with the MRC then in 1993, when I joined the MRC as a postdoc fellow, and started working on the immunology, because they wanted to exploit my expertise in immunology to study viral infections. At that time, there was a measles outbreak, and we did quite a good number of studies there about measles outbreak in the Gambia and how the body responds to it. And I then proceeded, after 5 years, we got good publications out of that, and then moved on to study HIV infection.
Dr. Jay, perhaps maybe we should take it a little more slowly.
Okay.
So that the commissioners and the audience would digest the information.
Okay.
Because this is information overload. and may I also apologize, Mr. Chair, for the lack of interpretation at this stage, because it would be mighty difficult to interpret this in any of the local languages. So perhaps maybe when we go to the gist of the issues, we would have an interpretation in Wolof. Not, of course, for Dr. Jay, but for the audience that is listening at home.
Okay.
Proceed, please.
Yeah, so I then did a lot of work. I mean, I moved from my research from measles to HIV infection, and then set up the measles immunology group, and we became eventually one of the strongest HIV immunology group in the whole world. The reason was because in the Gambia, we had the opportunity to have two kinds of infection, both HIV-1 and HIV-2 infection. And the HIV-2 infection was a bit rare in other countries, except in the Portuguese colonies, such as Guinea-Bissau and others. And so we developed a cohort, a very sizable good cohort of HIV-2 and HIV-1 infection in the Gambia and in Guinea-Bissau. The reason was that the HIV-2 infected individuals are long-term non-progressors, and we wanted to understand why these people are not as affected as HIV-1. Okay.
But this major breakthrough discovery was done in Gambia.
These studies were done in the Gambia, and the discoveries where we had actually identified that there were certain components of the immune system that are responsible to fight initially against HIV were discovered in the Gambia. And our group, because we had that that advantage to compare an infection that do not progress well with an infection that progresses, we had that opportunity. we were able to inform those who work in HIV-1 in other parts of the world about what is really happening in terms of the immune reactions. And by the way, about the qualification, I think one of the reasons why I was elected as the first government to be elected in the African Academy of Sciences was because of such a work we were doing. So I became a fellow of the African Academy of Sciences.
Congratulations. The first Gambian to be elected in that body is quite a lofty achievement. But you did not only register that kind of success in research, even your undergraduate studies, you did receive a number of awards, didn't you?
Yes, that was the reason why I was immediately offered this prestigious scholarship to do PhD, because my external examiner felt this was exceptional. And yes, during my Doctor of Veterinary Medicine degree, I continued to receive, during my period of that course, I continued to receive awards and prizes, academic prizes.
But apart from all that, you occupied important positions at your place of work at the MRC.
Yes, I started as a postdoctoral fellow. Out of the good work that we did in measles, I was promoted to become a senior postdoctoral fellow. And then for another 3 years. And then after that...
And that position was funded by?
That position was funded by Rockefeller.
The Rockefeller
Foundation.
That's right.
And then after that period, when we successfully worked on measles and produced some good results, and one of the important outcome of that work in measles was actually to show WHO was actually recommending 9 months, and you cannot vaccinate again, or you cannot vaccinate before 9 months of measles. And we wanted to prove that it's possible. And I had to do some, I don't want to go into the scientific details, but we had to go through certain real rigorous laboratory work to prove that, the passive immunity does not, is important, but does not prevent a revaccination. So we published this, and now that became also an information for the WHO to say, to build herd immunity, you can actually do a 2-dose vaccination in Africa. And that's why where there are outbreaks like currently in DRC, I think in Congo, they are now trying to do double-dose vaccination despite the 9 months schedule.
And that was as a result of the success of your research?
That was as a result of our work, and the publication is actually indicated in the CV.
That's right.
Yeah.
So as a result of that, you got promoted to
I
start
then became
working on
an international scientist, appointed directly by the MRC office in UK as an international scientist.
The MRC office in UK is part of the London School of Hygiene and Tropical Medicine.
That became recent. During this period I was working, it was independent. It was basically run and funded by, it was funded but also oversight administered by the Medical Research Council in the UK.
In the UK.
It's just about 2 years ago that MRC UK decided to merge us with the London School of Hygiene and Tropical Medicine, and now we are part and parcel of London School of Hygiene and Tropical Medicine.
And that is, well, it just speaks volumes about the credibility of MRC.
Exactly.
Thank you very much. So in 1998, you became a scientist specializing in immunology.
Yeah.
And you became in charge of HIV and measles immunology.
Yes.
And after that, what did you do? Let's talk about your career progressions, and then we would come to other aspects of your work.
During that period, actually, my major interest was really to understand why people in HIV-2 infected, those who are infected with HIV-2, why they don't progress as fast as HIV-1. And I don't know whether you want me to really explain the differences between these infections.
We would come to that at a later stage,
yes.
Because we would want to examine how you progressed in HIV treatment in the Gambia and what was the ultimate objective to phase it out completely from this country.
To understand the treatment issue, we need to I need to introduce really some aspects of the HIV
Please go
itself.
ahead. Please go ahead.
HIV virus, as you know, as you're asking about my progression there in research, the HIV virus, people have to understand that it was discovered in 1980 or 81, around there at that time. And it's a virus that is special. It's in the family of retroviruses, but it's a special retroviruses in the lentiviruses because it has a two positive strand RNA in its genome. And it jumped from chimpanzee. For the HIV1, it jumped from chimpanzee to humans, whereas the HIV2 jumped from Sooty-Mangabe monkeys to humans. Now, it was believed that this chimpanzee one originated from Central Africa, and the Sooty-Mangabe SIV, simian immunodeficiency virus, which is its origin, jumped from monkeys to humans in Guinea-Bissau. And that's why when during the Portuguese War and the exchange of soldiers, that's why the HIV-2 became very spread in the Portuguese colonies through that. Now, this work on molecular clock actually confirmed that this jumping perhaps started in 1931 for HIV-1, jumping from chimpanzee to humans, whereas HIV-2 was about 1940. But that's the history of it. And the thing about the HIV-2, why it is special, is because the two RNAs have the capacity to integrate itself into the human's DNA itself.
Now, this is very interesting because this is at the heart of the issue whether can be a cure for HIV,
exactly,
or what you can only have is treatment.
Exactly.
But let's go back to your qualifications.
Yes.
I just want to show how much of an expert you are in this area.
Yes.
I know you're much more excited talking about the technical issues than really showing your qualifications, but we have to go through this.
Okay, no problem.
All right.
Yes. Okay.
So in August 2008 to January 2011, you became the interim head for the viral diseases program at MRC, correct?
Yes, yes.
And in January 2011 to March 2016, you were senior scientist and head of HIV research, the West Africa collaboration, and you were seconded to Laboratory of Bacteriology and Virology
yes,
at Cheikh Anta Diop University in Dakar, Senegal.
Yes.
So pretty much for most of your career, you are a researcher on immunology.
That's right.
And you've made quite a number of publications in this particular area, haven't you?
Yes.
You have a copy of your CV, do
yes.
you? Could you take a look at it? And give us a brief rundown of the publications that you have made. I know that looking at the CV, it's by the way, Mr. Chair, it's about a 13-page CV, 12 pages. You've made about 85 publications in international journals.
That's right.
Could you just run us through just a selection of the publications? We would exhibit your CV. Just give us a brief run-through of some of them.
Of some of them, okay.
Yes.
Initially, on the measles vaccine, on the measles work, you could see that publication 5 and publication 6, where I was the first author, they were really showing good work on how the responses are when you get infected measles. But publication 6 for that was a very difficult journal to get published in. Only very, very high quality science goes into this journal. So for
what's
HIV,
the name of the journal?
Clinical Journal, Journal of Clinical Investigation.
Okay.
If you will be a very lucky scientist to get a publication in this journal. So, but for the HIV itself, actually it flows from publication 9 to, onwards, But if I give specific examples of where we really made good inroads and provided information internationally, would be publication 17, where, first of all, we discovered that the quantity of responses between the two infections are the same. But the ones that later confirmed, that also informed the international community was where we looked at quality of responses, and publication 23, where the first author was my PhD student, published another arm of the immune response to HIV-1 that shows a quality of immune response in HIV-2 different from HIV-1. And it also follows in 25 the same kind of similar kind of work that really showed that there is some quality of responses in HIV-2 that can inform HIV-1 vaccine development. That's also my PhD student. And it follows similarly into 26, 20, 30, publication 30, and also 32. All these ones were work that we vigorously did here to really show, to inform the world about the nature of the immune responses to HIV infection.
And that was published on the European Journal of Immunology.
Yes, exactly.
Proceed, please.
Yeah, so it goes on like that, actually. I don't know whether I should really give, but, there's quite a number of the publications that are, and in terms of treatment and resistance, multi-drug resistance issues in HIV, publication 37 was done by one of my students also there in page 37, and also 36, where it shows the responses of the heart in the HIV-1, HIV-2 dually infected patients in the Gambia, and the emergence of drug resistance. We published that in Journal of Clinical Microbiology, and a student who is a Gambian PhD student there did that work under my supervision. And equally, we've published the nature of the virus in HIV-2 itself structurally, and that's publication 39. We also wanted to understand what is the quality of the function of the response when you are co-infected with TB, and that publication is reflected in for page 44. Yeah. So there are some epidemiological studies we did to really understand the natural cause of the infections in both Guinea-Bissau and the Gambia, and that's reflected in publication 47. And I was particularly also interested because I set up the genetics lab called the HLA lab, Human Leukocyte Antigen typing lab. We wanted to understand the influence of the genetics to the response of HIV, and a student of mine did some work there to show the influence of HLA class 1 and genotypes for HIV on HIV-1 2 infections to see whether that influences the progression, and that you could see in publication 48 by the so I don't know whether I should continue because the list is so long, and I thought you wanted some examples on it.
We want to appreciate the extent of your involvement in research in this area. Yeah. We want to know the extent of your expertise in this area.
Yes.
We want to know, we want to have a good foundation upon which we would evaluate the opinions that you are going to offer to this commission?
Yeah. So no, certainly it's all given by the quality and the variability of the kind of scientific work we've done, including the immunology, including the genetics, including epidemiology, and also including clinical treatment and of both HIV and co-infections. And it's all reflected in the various publications listed here.
And Dr. Jaye, these publications are in the most valued medical journals in the world.
Yes, these are all internationally recognized medical journals where it's peer-reviewed. It's that one of the difficulties in research career is getting your work accepted in a publication. It's a challenge. You don't just submit and get it accepted. It's peer-reviewed, and there are criticisms. Sometimes you go through lots of problems before these papers are accepted, and the published journals are ranked into qualities. They call them impact factors, and all these journals, most of or three-quarters of these journals are actually at a very high impact factor.
And you have published about 85 articles.
Exactly.
But apart from publications, you have supervised researchers at the highest levels.
Yes.
Could you give us a few examples?
The number of students I've supervised?
Yes.
I think I've listed them in my publications. There's so many. I have supervised a Gambian for master's program at Ulcers called Jibrilba. I've supervised Louis Marie for a master's program. I've supervised Victor Nouveau, a Ghanaian, for PhD, which he submitted in 2009. I supervised Abdurrahman Haman from Ghana on an MPhil program and worked on mycobacterial antigens. And in 2008, I supervised Irfan Zaidi, who was from Pakistan, and he worked on HIV-2 Trrc infections, and he submitted his PhD in 2013. I supervised Alberta Davis, a PhD student with a Gambian, do a PhD, currently actually still doing her PhD. I've also mentored and supervised Dr. Sabel Jallo, who is now a postdoc working in South Africa. I'm still her mentor. I was an advisor in the PhD. I supervised a master's. I mentored Dr. John McDermott from UK. I also mentored Dr. Odwak when she was in the clinic working on a treatment of HIV, and Dr. Togun, who is currently a postdoc in London, at the London School in London. I mentored him when he was a physician responsible for treating the HIV patients. And Dr. Mustafa Mbow from Senegal, I'm still his mentor, and we recently, just 3 weeks ago, published one paper in Science. He's a Senegalese, and he's a postdoc working at Cheikh Anta Diop University. Dr. Moussa Cham also is a postdoc who is my mentee, and still in Senegal working on HIV infections. So these are some of the, I've only listed the others that I'm a co-supervisor, but these are the ones I directly supervised.
But you're also a member of several high-profile committees, such as the Association of University Teachers in the UK.
Yes.
You are also a member of the Royal Society of Tropical Medicine and Hygiene. You're also a member of the Gambia Vets Association.
Yes.
The Gambia Immunology Society.
Yes.
You are a board member of the board of...
I'm the president of the Gambia Immunology Society.
Fantastic. West Africa Livestock Innovation Center, you're also a member?
I'm a member of the council.
Yes. And a board member of schools and so forth.
Yeah.
Okay. It's a very fat CV, Mr. Chair. I just ask that we put it in, we exhibit it, and the commissioners will take a look at it later if they are interested. But clearly, it shows the depth of knowledge, extent of experience that Dr. Jaye has in this area. Mr. Chair, I had asked that the copy be circulated to all commissioners so that they can follow as we progress. I wonder whether you have it before you. Okay. I ask that it be given exhibit number 157 if it is accepted, Mr. Chair.
Request granted, Council.
Thank you very much. Dr. Jaye, wealth of experience in immunology. Now kindly give us a brief history of the discovery of HIV. I know you've told us how HIV-1 has mutated from chimpanzees around the Congo and HIV-2 from monkeys in Guinea-Bissau in 1931 or thereabout, and ultimately their discovery in 1981. Could you please take it from there, please?
Yeah. Shortly after the discovery of HIV in 81 in the whole world, which became really a global epidemic, Gambia discovered some individuals with HIV through epidemiological surveys that they've done. I hadn't then started work on that because that was around 83. So it was clearly very early done by one of my mentors, Professor Hilton Whittle, who's now retired, who really started it. They built up a cohort in Guinea-Bissau, small cohort. They realized the HIV-2 was really there, but then was also in the Gambia. And I joined in 1993 when that cohort was beginning to develop. We then developed a cohort after I joined the HIV group in 1998, after following my measles work. We developed the cohort, which grew up to about 1,600, comprising of about 800 HIV-1 and 450 HIV-2, and the rest were dual infected people. Dual infected people means being infected by both HIV-1 and HIV-2.
During this period, could you just give us a brief rundown of the discovery of medicines for HIV treatment?
Yes, so initially, as I said in 83, the only work, we confined our work to understand the prevalence of these infections in the Gambia and how problematic it would be. Then following the setup of the immunology group, treatment just started when the first drug was discovered, I think around 1987. The first drug that was discovered was called AZT in short. This is zidovudine. And this was the first drug. And when it started, we immediately started using the drug for HIV-infected individuals. Yeah? But at that time, it was just a clinical use. It was found not to be quite effective, the single drug, because what happened is that there was quite a good mutation. And let me just go back and say the two major problems of HIV is because I mentioned about the ability to integrate into the DNA. But if it integrates into the DNA, it has that ability to hide itself, for a long period. And always, that's the reason why it's always one step ahead of the immune response of the body itself, the soldiers. The soldiers responsible for doing the surveillance and attacking. And the second thing is that when it comes out in bits. It has the ability to mutate. Mutate means it changes some of its sequences. So when you give a drug and recognize the drug, it tells other parts of him to say, go back and hide. And then when it comes back, it will have changed some of its sequences that the drug will not be able to attack. And so that single drug was ineffective because of that.
But before this single drug, HIV was basically a death sentence.
It was a death sentence. It was a death sentence. And when even that drug came, it was still a death sentence because of these problems that I'm now alluding to. And that's why I want you to understand that the major problems of HIV, which to some extent is still existing. Now, beside that changing itself, it has the capacity to multiply at a very high rate. But with that multiplication, it always changes its sequence, because what really dictates its multiplication is called a transcriptase, transcriptase enzyme, which actually makes it to multiply. But then the way it's doing that is error-prone, and that's why it's easily able to mutate and change. And so that high diversity becomes an issue in HIV. So I just want to mention why the treatment is a problem, because of those factors and that capability that HIV has. Now, yes.
And because it was so difficult to treat, it was a death sentence.
It was a death sentence.
Everybody was scared of it.
Everybody was scared of HIV. And once you get it, you just think that, I mean, the chances for you to die is very high. And even with the zidovudine discovery.
Yes. But because it was a sexually transmitted disease, the stigma attached to it was also very high.
Very high.
And this resulted in this dual problem of contracting a sexually transmitted disease that was definitely going to kill the person.
Yes. Yes.
That created a problem.
That created a problem. So few years later, and this zidovudine is under the class of what they call nucleoside reverse transcriptase inhibitors, because these drugs, what they do is that the virus depends on some enzymes to integrate and also to multiply. And these enzymes are called reverse transcriptase. Then there is a protease, which enables it to, to also get that machinery of the host cells to multiply. There is the integrase enzyme as well. So these drugs are designed to inhibit these enzymes. Now, zidovudine inhibiting the transcriptase enzyme so that it won't multiply was not quite efficient enough, because the virus can have an escape route to multiply. Then all the drugs came later to be discovered in the same class of nucleoside reverse transcriptase. And one of them is called Trrc or lamivudine. Now then and that was where our first trial came, our first treatment trial came, when the second drug was discovered, lamivudine. So we thought, and that was what was recommended then, that if perhaps we combine the two drugs, it will help, it will help the patients to a level that there won't be mutations and, drug resistance. We then did what is known as the viral turnover studies. I think it was around 1998, if I remember, when we started that, to do this two combination of drugs. But they are the same class in the nucleoside reverse transcriptase class, and they call it Combivir. We did a trial that was approved by the Gambia Ethics, Joint Gambia MRC Ethics Committee, because any research you do in this country, including clinical trial, has to be first approved by the ethics committee in this country.
Why is that the case?
Because there is no research, medical research internationally, that's supposed to be done anywhere in the world without approval from an ethics committee. And the ethics committee's role is to protect the patient, not the researcher. So they have to look at the ethical issues. are these how you give the drugs, how you follow these patients, and what are the actual methodologies you use, and how are you disadvantaging the patients you've recruited, and what is the recruitment procedures, and whether these patients have given informed consent. These are the ethics issues that any ethics committee must scrutinize and look at properly before they give approval.
It looks to me that the entire practice of medicine is anchored on some significant ethical considerations.
No, yes, but not the treatment, not medical practice and treatment. No, you don't have to have ethical approval for that. Routine medical data and clinical routine treatment, you don't. But research, research and clinical trial, you must get an ethical approval.
That is understood. But my question is even broader, that my proposition is that the entire practice of medicine is anchored on some strong ethical foundation, which should always be observed by the practitioners, be it research or actual practice.
That is true. That is true.
At this stage, could we have interpretation in Wolof for what can be interpreted? And, or we just proceed in English? Because I'm wondering how much of this could be proceed in English,
Please
Mr. Chair.
proceed.